So I'm Divyanshi. I'm a graduate student in the lab of Dr. Sean Mahoney, and this is work done in collaboration with Milica Bulacic and the Mazzoni lab at NYU. So today I'll be talking about our work on how Hox transcription factor binding specificity is directed both by DNA sequence and by differential abilities to engage inaccessible chromatin. So the Hox genes are an evolutionarily conserved family of genes that are extremely important during embryonic development. The Hox genes encode for transcription factors, each of which become expressed sequentially along the anterior-posterior axis during embryonic development. And the expression of each Hox transcription factor results in the development of a specific body part or a specific positional identity. So as an example, shown here is the organization of Hox genes in Drosophila, as well as in vertebrates. So interestingly, despite specifying extremely different positional identities, the Hox transcription factors bind very similar DNA motifs. So the anterior Hox transcription factors, HOX1 through HOX8, bind an anterior TAAT motif, whereas the posterior Hox transcription factors, HOX9 through HOX13, bind a posterior TTTAT motif in vitro. And therefore, given this similarity in DNA binding preferences, it is not clear as to how the Hox transcription factors can specify distinct cellular fates and distinct positional identities. So this will be the question that we'll be trying to answer through our work. In order to characterize Hox transcription factor binding in the genome, we decided to focus in on motor neuron progenitors. So motor neuron progenitors exist in the developing spinal cord. And within these progenitors, individual Hox transcription factors become expressed, resulting in diverse motor neuron subtypes. So the expression of HOXC6 results in the formation of brachial limb targeting motor neurons. The expression of HOXC9 results in the formation of thoracic targeting motor neurons. And the expression of HOXC10 results in the formation of hind limb targeting motor neurons. So what we did was, so let me backtrack here for a second. So what we expect is that we expect HOXC6 to bind an anterior motif, but we expect both C9 and C10 to bind similar posterior motifs, therefore making it unclear as to how they're specifying differential motor neuron identities. The system that we used to study this was motor neuron progenitors derived from mouse embryoid bodies using a differentiation protocol. Once we had these motor neuron progenitors, we used DOX-inducible Hox constructs and expressed individual Hox transcription factors within these motor neuron progenitors. So we induced C6, C9, and C10, and 24 hours after Hox induction, we measured the genome-wide binding of these three Hoxes using ChIP-seq. The first question we asked was, how do these transcription factors bind in the genome? Interestingly, we find a diverse binding profile for all of these different Hox transcription factors. We find a set of sites that is shared between the Hoxes, but we also find sites that are uniquely bound either by HOXC6 or by HOXC9, as well as subsets of sites that are bound by HOXC9 and HOXC10. Looking at the motifs that exist at these different categories of Hox binding sites, we find, as we expect, that HOXC6 unique sites are associated with an anterior Hox motif. This is in line with what we expect HOXC6 to bind. However, sites that are bound only by HOXC9, as well as sites that are bound by C9 and C10, contain very similar posterior TTTAT motifs, and therefore it looks like sequence alone isn't responsible for driving differential C9 and C10 binding. To dig into this further, we used a discriminative K-mer model to look for whether there were more subtle differences in sequences at C9 and C10 binding sites. So on the x-axis here are the different binding categories, and on the y-axis are different motifs that are derived using a differential K-mer comparison. And what I want to focus on here, down here, is that we find no discrimination between motifs at C9 only sites as compared to both C9 and C10 sites, suggesting that the diversification in C9 and C10 binding is not driven by differential sequence preferences. So having established that sequences may be not playing that important a role in driving differential C9 and C10 binding, we next asked whether the prior chromatin landscape could be responsible in influencing the genome-wide binding of these three Hox transcription factors. So in order to do this, what we did was we took these motor neuron progenitors, and before the induction of the Hox factors, we measured the chromatin environment within these progenitors. So we used a TAC-seq to measure chromatin accessibility, as well as ChIP-seq to measure a bunch of different histone modifications. And we asked whether this chromatin landscape predicts induced Hox transcription factor binding. So in order to do so, we developed a computational approach that can integrate both DNA sequence information and pre-existing chromatin information, and use these two data types in order to predict transcription factor binding. So let me walk you through this, this model. The idea here is that we first have a sequence subnetwork, which I'm calling bichrome-seq. This network takes as input one hard encoded DNA sequences. It uses a fairly standard CNN LSTM and predicts and outputs a single real valued activation, which I'm calling A sub S here. The chromatin subnetwork actually acts fairly similarly. It uses a CNN LSTM structure. However, instead of using DNA sequences as input, it uses pre-existing chromatin data as input. And now both of these activations, A sub S and A sub C, are weighted by a final sigmoidal node, which is then used to predict transcription factor binding. However, what this setup allows us to do is, for each individual binding site in the genome, it allows us to estimate how much of the prediction is coming from the sequence network and how much of the prediction is coming from the chromatin network. In other words, it actually produces this two-dimensional latent embedding, capturing the contributions from sequence and chromatin at each individual binding site. So we applied bichrome to HOXC6, C9, and C10, and I'm showing you results here for HOXC6. In purple are the HOXC6 binding sites. And very interestingly, we find that HOXC6 sites occupy diverse regions within this two-dimensional latent embedding. There are some sites here which are very strongly predicted by the sequence, but we also find sites up here that are weakly predicted by the sequence subnetwork, but very strongly predicted by the chromatin subnetwork. We find that the chromatin scores assigned by the network actually correlate with active chromatin. So chromatin that was previously accessible and contained K4 trimethylation and K27 acetylation is assigned high chromatin scores, whereas chromatin, which was previously quiescent and showed no enrichment for any histone modification, is assigned a low score by the chromatin subnetwork. So given this, this compensatory pattern which we observed, we wanted to ask whether the sequence motifs at these previously active sites differ from the sequences at previously quiescent sites, because we can see that previously quiescent sites are typically assigned these high sequence subnetwork scores. And we do that here. So I'm showing you results again for HOXC6 and the enrichment of various primary and secondary motifs at the sequence predicted sites bound in quiescent chromatin versus chromatin predicted sites. bound inactive chromatin. And what I'd like to focus on here is the primary motif, and we see that the primary motif is enriched at a higher frequency in these sequence predicted sites, suggesting that a higher primary motif enrichment may allow for Hox binding in previously quiescent chromatin. We also observe differential enrichment of a whole set of other secondary factors. We find that the same pattern holds for both HOXC9 and HOXC10. We find a greater enrichment of the primary motif, so the TTT8T here at sequence predicted sites, which are more likely to be bound in quiescent chromatin, as opposed to chromatin predicted sites, which are primarily bound in open chromatin. So given that we can analyze individual binding sites, and we know that the chromatin seems to be predicting to some degree the binding of all three Hox transcription factors, we next asked if we could go back to the network and use this network to interpret what the total amount of information coming from the chromatin is. So in other words, we wanted to ask how much is the pre-existing chromatin contributing toward transcription factor binding at a global level. In order to do this, we did something very straightforward. We used the sequence network, and we asked how well does it predict induced transcription factor binding. We then used the full bichrome model incorporating pre-existing chromatin information, and we asked whether this whole network actually performs better than a sequence-only network at predicting induced binding. If it does, that would suggest that the pre-existing chromatin data is predictive of induced transcription factor binding. So I'm showing you the results for this comparison here for HOXC6, C9, and C10. And very interestingly, we find that HOXC9 behaves differently from HOXC6 and HOXC10. So both C6 and C10 gain a substantial amount of information upon the incorporation of prior chromatin, but HOXC9 does not gain as much information when we incorporate prior chromatin to predict Hox binding, suggesting that HOXC9 might not be dependent on pre-existing chromatin to the same degree as HOXC6 and HOXC10. So taking this insight, we went back to our Hox binding sites, and we plotted the chromatin accessibility at these sites prior to Hox induction. And interestingly, we found that sites that were bound by HOXC9 only, but not HOXC10, despite containing the posterior motif, were extremely inaccessible prior to Hox transcription factor induction, suggesting that it is an ability to bind inaccessible chromatin at these set of sites, which might enable diversification in C9 and C10 binding. So finally, having looked at the binding of HOXC6, C9, and C10, we wondered whether additional posterior Hoxes also behave differently in their abilities to bind inaccessible chromatin. We first looked at HOXA9 and D9. So these guys are direct paralogs of HOXC9, and we wondered whether they behave in a similar manner as HOXC9. We again ran both a sequence-only network and bichrome to predict the binding of these Hoxes. And interestingly, we find that despite being paralogous to HOXC9, both of these transcription factors gain a substantial amount of information from the pre-existing chromatin data, suggesting that they are more dependent on the chromatin landscape, and C9 is unique in its ability among these transcription factors to engage inaccessible chromatin to a greater degree. Finally, we also assayed HOXC13, which is a posterior Hox transcription factor, and we find that similar to HOXC9, the addition of pre-existing chromatin using bichrome in green here results in almost no gain in predictive capacity, suggesting that HOXC13 is not dependent on the pre-existing chromatin landscape. And this result ties in very nicely with a recent paper that was published establishing HOXC13 as a potential pioneer transcription factor. So with this, I'd like to conclude my talk. We think that in addition to sequence, pre-existing chromatin can allow for diversification of Hox transcription factor binding specificities. And this intuition is captured nicely here in this genome browser snapshot, where we see HOXY13 and HOXY9 bind deeper into previously inaccessible chromatin, which contained low attack-seek and high K27 trimethylation signals. So thank you. And with this, I come to my acknowledgments. I'd like to thank Sean for all of the help with this work, everyone in the Mahoney lab, as well as everyone in the Mazzoni lab. And finally, I'd like to acknowledge our funding sources and thank you for your attention.